Thank you very much for that, Jim and Dennis, and thanks to all of you for coming here. Listen to a northerner talk to you a little bit about a problem that I know many of you are quite familiar with. I am going to right away, though, try to clarify some terminology. If we talk about red tides down here, this is an image that I think would sort of resonate with many of you. This is a picture of the types of red tides you get down here. This one's in the Gulf Coast of Florida. You can see the water is brown and discolored because of this organism we're going to talk about, and problems you might have troubles walking on the beach because of aerosols and so forth. There may well be dead fish and so forth. And this is what people down here call a red tide. If you went up north and you took a look at this body of water, the local papers would say the red tide strikes Cape Cod again. And yet if you look at it, the water's not red. There's no problem breathing the air. But if you looked very closely at this sign way over here, it would tell you, warning, do not eat the shellfish, because if we harvested some clams right from here, the day I took this picture, just one or two of the clams would kill any one of us in this room. They're that toxic, that poisonous, even though the water is blue and seemingly harmless. So that's why, one of many reasons why many scientists don't really use the red tide term unless I use it in titles of my seminars because it brings people out. They know what I'm talking about. But if you're talking with scientists, we use this term, harmful algal blooms, or HABs, or HABs, because it's a much bigger term. And I'm going to give you some slides. I'm going to spend a little too much time on this in the beginning, but after a lot of discussions today, I realized that I want to cover this general topic of HABs and red tides a little more broadly to get you a feel for just how diverse the phenomena are. Then we're going to go back and study in detail some of the issues we're using in the Northeast, but I want to give you the broad perspective first. Now, if we went back to your bloom that I showed you and looked at it under the microscope, you'd see this. A lot of individual cells. These are tiny little plants, microscopic plants that photosynthesize. This would be Karenia brevis, the organism that causes the problems here. This would be the version of the water in my neck of the woods. In this case, this little golden ball is the organism that causes us most trouble. It's called Alexandrium. Now, I show you this image for two reasons. One is to first give you the feeling of how Alexandrium looks in the field, but also to point out that this is the assemblage in which these organisms typically occur. They're rarely the only thing in the water. And the reason that's important is for those of us trying to study them, to measure them, to learn their dynamics, to learn their toxicity, all of this is basically noise in the system. We care about what this cell is doing, how many there are, how toxic they are, but all of these other organisms are there as well. It's like me trying to do a study here and figure out what's the pulse of all the people who are from Massachusetts in this room. Very, very hard to do with such an assemblage. And that's what we have to do in this field. And that's where some of the challenges with this biosensor technology comes in. So let's go back to well, that's right, I forgot. For those of you who don't like looking at the water, this is the same thing on land. Imagine each one of these is a different species. They're blooming. We talk about algae blooming the same way. And there's many, many, many different species here at the same time. And imagine that the only one you really wanted to study because it was poisonous somehow was this orange one. And that is your challenge. So the basics of this organism's life start here. Every one of these cells that we're talking about starts as a single cell that then divides. As they grow, they don't get big like a tree. They grow and then they divide, and then they grow and they divide again. So that's why you go from 2 to 4 to 6 to 8 cells, and that is the population that typically can change the color of the water or can make things dangerous. Now, to give you a few examples, if you get an organism like this one, it may look spectacular, but it's actually harmless. In this particular location, offshore, this is a non-toxic organism in New Zealand. This is true color. There's nothing fake about this. It really does look like tomato soup. But if all of this biomass, as we call it, gets blown into shore and gets real close and starts to break down and decay, it uses up oxygen, and when that happens, it basically kills everything that's there in those shallow waters. So you have one effect of non-toxic HABs that kill things just because of oxygen depletion. But most of the problems we deal with are things like this. This is, again, the Florida red tide. The water is typically reddish and brown. Now you see a lot of dead fish floating in there. These are fish that have died from a very potent neurotoxin that is produced by and released by that red tide organism and that kills the fish. So this is not an unusual sight for some of you. I'm sure millions and millions of dead fish along the coast. So these toxins can also now kill fish. Now here's a very interesting example. This is where I stuck in a few slides, and I know I'll regret it time-wise, but it's a fascinating story. The farmed fish are also killed by some of these organisms. It's actually, some of them are being killed by a different toxic mechanism, but I won't go into that. But what you can see here is that very recently, in 2016, in Chile, almost $1 billion worth of salmon died in just a few weeks because of a red tide down there. And I was down there, and I just came back from there again. You can see these huge farms. They have many, many, many of them. They're massive in scale, and a good half or more of the salmon that you might eat will come from these Chilean farms. And what you see, though, is that the fish were dying from one of these blooms, just so many dead fish. And I just will show you a few pictures because the fishermen, the farmers were so handcuffed by so many dead fish that they dumped them out at sea. They got permission from the government to dump them at sea. And what happened was that another red tide, a different species, appeared, shut down the shellfish this time, making the shellfish poisonous, and it triggered a true social protest, a social uprising. And this is a real picture, as is this one. Basically, they barricaded all of the ferries. They barricaded highways for 3 weeks. They burned tires in the street. This looks like it could be a riot in one of our inner cities. This was in Chile as a result of fish kills caused by one of these types of algae. So it's just a striking example of how this phenomena can affect society. Now let's get back to the impacts. A number of these problems that we call HABs show up in shellfish. This looks like a nice plate of mussels, but mussels and many other of these bivalves will filter the water, filter algae from the water as food, and then accumulate the toxins to the point where they can kill you or you can get very, very sick. So you've got a shellfish poisoning. You've got seabird mortality, sea lions, whale mortalities. Many, many of these monster animals now we're finding out are being killed by the toxins produced by these tiny, tiny organisms. And then this, just to show you how broad the term is, these are pictures I got years ago from Brian LaPointe right here, showing overgrowth of seaweed here, this codium here in the region, and I think that's Brian standing in up to his waist in this seaweed. Seaweed is algae also, so this also comes under that term harmful algal bloom. But just to show you, people are asking me, well, I was just in China, why was I there? Well, this is one of the reasons I was there. In 2008, they had a seaweed bloom that has now continued every year since then that you will not believe in scale. This was at the Olympics. This is one of our windsurfers or sailors trying to sail in this incredible green tide, and it was so massive that the government had to bring in 10,000 soldiers, many of which you see here, a lot of people from the city of Qingdao, and I'll just show you some pictures to show you the scale of this phenomenon. This is just local citizens trying to help cleared off the beach. 100 million tons of algae washed up in that part of China, and something near that has happened every year since then. That is an amazing story. We can talk about later why it's happening. Believe it or not, every time you eat sushi and you eat that little nori that wraps around it, that ultimately is the cause of this Chinese green tide. But I don't have time to go into it, so I'll just leave you with that teaser. But you can see the incredible, and this is the same ulva that I saw today as we were going around looking at your aquaculture facility. This is being grown and can be eaten, and they're trying to figure out ways to do it, but in this quantity, all they did was throw it away. So just to wrap up all this background, I want to make sure you realize that there's also freshwater equivalents of what I've shown you in the ocean. This is a picture of Lake Erie and a bloom of an organism called microcystis that you can actually see from space. And for many of you, you might remember this 2014 when basically Toledo, Ohio shut down its drinking water supply for several days because of the toxins produced by this organism, and the plant was not ready to remove them. And so the people there went without water for a number of days because of it. So this is just a list of a number of HABs and their impacts, and I highlighted in yellow all the ones that occur down here in Florida in one way or another. So this is really an amazing state for HABs. I've got a lot of good colleagues working here for good reason, because you have many of these poisoning syndromes. You've got at least 3 of them. You have a fish poisoning syndrome that is something that's largely associated with reef fish, the large reef fish, especially down further in the Keys, and then brown tides, you're having here in the Indian River Lagoon and so forth. So it's a big, diverse problem. And of course, those of us studying it are always trying to find better ways to study it, better ways to manage it. And that then gets me to the main part of this talk. And if I wanted to talk about new technologies for HAB research, there are many, many, many. I've just listed a couple here. There are things like new methods for detecting the toxins, new quick methods like pregnancy kits for using this, or much better analytical methods, methods to even control the blooms, to try to suppress them. And there's many, many other technologies that we're working on. But what I'm going to focus on are these 2. New sensors to try to look at, to count and measure toxins in situ. It means in place, like within the water, as well as numerical modeling for forecasts and predicting these blooms. And these 2 I have to show them because they fit together. Some of the rationale for doing this work is because we want to do that. So now we're heading up to the north, although you do have this problem here in the Indian River Lagoon. This is paralytic shellfish poisoning, or PSP. It's that disorder, as I showed you, that could kill you if you eat shellfish that are heavily contaminated. In our region, up here, Woods Hole is right there. This is called the Gulf of Maine. This is then Canada and the United States. And everywhere in purple, you can see areas where there are essentially annually recurrent outbreaks of this paralytic shellfish poisoning. So it's really quite a prevalent problem throughout that region. And this is then the region we've been studying within the U.S. portion of that. Now, it's also important to understand a little bit about the life history of this organism. This is the cell I showed you earlier, and we call it now Alexandrium catenella. There's 2 cells there. But this is also Alexandrium catenella. But it's more like a seed. Think of it that way, a seed of a plant. And this is the life history. So let's start with this seed, which we call a cyst, which germinates, produces a cell that then divides and makes 2. And as I showed you, those 2 that one makes 2, this one makes 2, we get 4, and then the bloom begins and you get a large number of cells just through that process that we call asexual division, just regular binary division. But then at some point, and it's typically when these cells run out of nutrients, they form gametes which fuse together to make a zygote, which then produces that cyst. And this is the life history of the organism, and it's hugely important in trying to explain and predict what happens with these outbreaks. And I put this slide here just to show you that one of the organisms that you have in the Indian River Lagoon, and it's also over on the west side of Florida, is called Pyridinium bahamense. It's a similar class same class as the organisms I've showed you so far. It has, these are the vegetative cells here. It makes chains like this. but it also has a cyst stage, just like the Alexandrium cyst that can germinate and cause and liberate blooms. So a lot of what I'm saying about Alexandrium and its cyst does have an analogy here, and a lot of the technologies, the approaches we're taking can be used here, and it's actually a lot of the discussions I had here today are about possible collaborations where we can share some of these technologies. So up in the Gulf of Maine, we have two types of blooms, as we call it, of Alexandrium. You can see here's Cape Cod, and here's Boston, and here's Nova Scotia. You can see this coloring is, these are the cells of Alexandrium that we've counted on cruises that stopped at every one of these. See these little lines and sampled, and then we've mapped out the cells that we've counted. You can see the scale of this bloom is incredible. It covers hundreds and hundreds of miles. It's all moving along this coast and around Georges Bank. That's a major type of the bloom we get up there. But this, you can see, is much smaller. Look at this road, look at this building. You can see this is what's called a salt pond out on Cape Cod. And this is another place where we do a lot of our work. It's also, the reason we do is because it blooms essentially every year with Alexandrium. And part of the reason it does is this very narrow and shallow inlet channel sort of keeps the cells in here. The cysts are in here, they germinate, they bloom, and most of them stay there without being washed out. But I will come back to that. So, how do we study these? Well, the first thing that I have to go through if I'm going to talk about sensors are modeling. You're going to be bored by it. No, I hope not, because modeling is really, really important in every part of our life. When you think of, and I'll come back to this, weather forecasting, they've had to do modeling of the atmosphere. That's the reason they can do forecasts. They have to be able to model the movement of the fluid air. Well, we have to model the movement of the fluid ocean as well as the organisms that are in it. And we use these models for looking backwards in time, for looking at what's going on right now, and for looking forward. So how do we do that? Well, we start with a hydrodynamic model. We'd call this a physical model because it's just the physics of this system. This shows you salinity up in that region. This is the Gulf Stream. You see very salty. See little eddies and filaments spinning off of it here through time. The blue shows fresh water coming down from the Arctic, the melting glaciers and rivers and so forth up here. These are rivers entering up here, the St. John in the Bay of Fundy, the Penobscot, the Androscoggin. So this physics, though, is very well modeled, very well characterized in these hydrodynamic models. But that's, in a sense, the easy part. The hard part is here. We have to put a biological submodel into that hydrodynamic model so that we can say, well, what happens to our organism? And so I cannot possibly go through all this, but what I'll say is that we start with a map of the cysts of Alexandrium. We go up and run cruises and count in the sediments all of the cysts that are there. And we get maps like this that show what we would call a seed bed here and a seed bed here. This would be almost like if you went out in your yard and could somehow map out weeds like some dandelion or something like that before they would actually pop. So you would know there's a seed bed here and some here, but not so much in between. That is the potential, the sort of area of the quantity of cells that might actually start a bloom. Then you need to know when do they bloom. And that's what this shows. We've done studies. This is all data from the lab that shows there's certain times of the year where these cells will germinate, and here's the next year, and here's the next year, here's the next year. And there's lots of times where they either germinate not at all or germinate very poorly. And if any of you are gardeners, you know that's true for a number of different kinds of seeds. But I won't get into that right now. Once you've got them germinating, you need to know how fast they're going to germinate, and that's what's shown here. You have to, once they've germinated and are swimming around, we have to know how fast they grow as a function of salinity and temperature and light, and we put all of that in this model, give them a little bit of swimming behavior, and I'm skipping by years and years of work, but the result is here. Here's a model of the year 2005. We took a cyst map in 2004, and we have actually the way the currents and the rainfall and all the conditions happened in 2005. You can see this is going through, now it's up into June, July, then it's going to start over again in March. So you can see that the bloom is beginning in the areas of those cyst seed beds that I told you, that it's being carried down the coast by these coastal currents and so forth. It's going around George's Bank, down here even to Martha's Vineyard and Nantucket. And this model is actually a very good representation of what truly happened. This was a year that the state of Massachusetts had a $50 million loss to its shellfish industry alone because of this outbreak. So we've got a model. What can we do with it? Well, we do many things, but the one I wanted to highlight here is this one, weekly forecast. So we do something a little bit like the 7 or 10-day forecast for weather that you get when you want to go golfing or something else. But how do we do that? Well, we use that model, we run it with the conditions that are happening in that year, and then we get a forecast that runs out 4 or 5 days forward, and we extend the model with that weather forecast. And so myself and a lot of interested scientists and managers in the regions will then get a message like this from the people who are doing this modeling, and it gives you this link, and if you click on it, we jump out to this web page, and there is not quite the radar map that you get on the weather these days, but a map of the Alexandrium bloom. And you can see here, this is 7, 8, 9, 10 June. So we're showing what's happening in June. I don't even remember what year this is. I think it's 2016 or 2017. And the managers can look at this and say, well, I'm worried about what's happening here. You can see all the cells are offshore. They're not being blown in. Or if a storm comes, in they come. But we give them this information every week. But there's a real problem. If we looked at the beginning of this starts in March or thereabouts. And we start running it. Here, let's see if we can see where it starts. We start running it forward, and we can't correct it. If something is wrong, for whatever reason, something happened that wasn't captured by the model just keeps propagating forward. So it's more like me saying, would you trust me if I said, see, I missed it, but we're in May already. Would you trust me if I said, I'm going to give you a weather forecast for what's going to happen in June, but I'll give it to you right now? You wouldn't believe me, and you shouldn't. So what is it, though, that allows the weather forecasts to be, jump back here, so accurate? How can we improve our forecasts? And the answer is to take a lesson from the Weather Service. These are their models, very similar to what I've shown you for the models that we have of storms moving through the ocean. These are the same sort of computer models. But why are they accurate? Well, this is why. Because all over the region, all over the country, they have meteorological stations like these here that are collecting data constantly. They're putting weather balloons up that are giving you this three-dimensional feeling for what's going on in the atmosphere. And all of that data is taken by the Weather Service. It's assimilated into those models. They adjust them, and then they run them forward again. And those models are constantly being corrected and then run forward. So that's what we are not able to do so far with our HAB models. So how can we improve things? Well, what we need are in situ sensors. We need sensors that are equivalent of those meteorological stations, but this time for Alexandrium cells and toxins that can provide this real-time data that can be assimilated into the model, thereby improving forecasts. And that's where we have been working on two of these, two instruments. This one's called the ESP. This is called the IFCB. I'll go through each one, tell you what they are. The first one, the ESP, is called the Environmental Sample Processor, or ESP, developed by one of my former students and now the head of the Monterey Bay Aquarium Research Institute, Chris Sholin. You see the instrument here. It's about the size of a trash can, and here it's being deployed in one form. But over here is what's important, is that this technology, it's like a laboratory in a can, and I'll show you how it works in a second. But what it can do is measure both the abundance of species, of HAB species, as well as their toxins. It measures them both autonomously, robotically, inside a can. And so I'll be quiet for a minute.
Life, and the search for it, is perhaps our greatest adventure. On Earth, the most abundant forms of life are those that we cannot see with the naked eye. 3 kilometers below the ocean's surface, microscopic life forms flourish in the liquids seeping from the Earth's interior. What are these organisms? How did they get here? And what impact do they have on our environment? Each year, massive blooms of algae render parts of our ocean toxic to humans and wildlife. Imagine if we had an automated detection network warning us when harmful microbes were present. As we further industrialize the ocean, what is the effect of microorganisms in our harbors and off our coasts? Is there a better way to monitor how we are impacting the planet? Fish farms are especially vulnerable to outbreaks of harmful algae and disease. What if we were instantly notified of changes to the health of the waters around aquaculture areas? The Environmental Sample Processor is an automated laboratory able to detect microorganisms in real time. Consider the mysteries this biological and chemical sensor is beginning to address. Our ocean houses life forms we know almost nothing about. Where do they travel? How is humankind impacting their environment? And how are they, in return, impacting ours? The ESP provides us with a model for how we might monitor the health of our ocean in the future and seek out new forms of life in the darkest depths of our world and in the farthest reaches of others.
So I wanted to show you in that last one, and I couldn't stop it in time, that one of the sponsors of that research was NASA, and they helped a lot in putting that video together, as you can imagine, because at the very end you can see what one of the reasons they were sponsoring it is that they would like to see this very same technology, this robotic laboratory in a can technology, head out to other planets for the search for life. And so they're helping fund research in our own oceans and looking down the bottom and under all these hostile environments to help advance that technology. But we've been able to apply it to these HABs. Now, how do we do that? We've had to develop a very robust mooring system because of the currents and the tides and so forth in our region. See, this is the ESP here connected to the bottom. Very strong surface expression of that. Here's the cable that stretches 2.5 times its length and still can conduct information up and down. Here's a few more pictures of, it's a big operation of a number of our ESPs on a ship being deployed. You can see that there's the ESP in its canister here, batteries there, here's the surface buoy. And to give you some data, here's an example from 2014 where we deployed 3 ESPs along the coast up here in New Hampshire and western Maine. And the ESPs were put very close to the stations where the state of Maine tests the shellfish flesh for the toxins, the PSP toxins. We wanted to see what kind of information we got right near those stations. Basically, we had a 6-week deployment. We had ESP measurements once per day, and we had measurements of the shellfish toxicity by the state every week. And this is the kind of data that we get from those instruments. Over here is the cell abundance, over here is the time, and over here is the measurement of toxicity in the shellfish. That is this red line here, done once a week. This line shows you the level at which it becomes dangerous to eat shellfish. That's a threshold or an action limit. So here is the ESP data. You can see how patchy the blooms are as they go by that moored instrument. That there's cells there's none, there's none. But gradually we get more and more cells. You see that in general it follows this pattern that's showing up in the shellfish, that there's low toxicity here, higher toxicity there. You also see some interesting times when we get rather big jumps in the cell abundance that may not have been picked up very much in this weekly data from the state. Here's another ESP deployed a little further up the coast. Again, you see the state data, the shellfish becoming more toxic. The quarantine level is, actually the quarantine level in this case is way up there. Here's the Alexandrium abundance following some general pattern. And we give these, just 2 examples of many, we give this data on a real-time basis to the managers so they can know what's happening. But it also occurred to me that these are useful, but how can we make them even more useful to the resource managers? And one of the answers there is that this is the currency they talk in. It's the toxin measurement. This is the currency that the instrument's giving them, which is cell abundance. They're not the same thing. They're related, but they're not the same thing. So what we've done is developed something, and don't go bleary-eyed here. This is really very simple. What we've done is make the ESP into a robotic muscle. We're saying, okay, instead of counting cells, well, count your cells, but let's use those cell counts and convert them to shellfish toxicity as if that ESP were a muscle. And this is a formula that it takes the toxin at the next time point and says it's equal to how much toxin we had at our last time point plus an uptake number minus a loss number. And this is, the uptake number is just some constant times how many cells we measure with the ESP. And this is a constant times how much toxin we had at the previous time point. It's real simple. We fit this model to the data from the ESP and the state agencies. And then this is the kind of data results we get. Now you see this says toxin over here on the ESP side, and it's toxin, well, it's toxin for both. And this is the state data, these circles, and now we see the ESP data that we are hitting those state results perfectly, almost, but we are also seeing all the dynamics in between. And as I said, between this point and this point, look, there was a number of days where our robotic muscle would have said there were dangerous levels of toxin that the state was not picking up with its weekly sampling that a sensor out there every day would have. We look at the other example I gave you. Again, an extremely good fit to the data, but also these kind of peaks that we're not necessarily seeing in the state data. And we've done this for a number of different years, and this is a rather remarkable result where we've got our modeled toxicity in the ESP plotted versus what's observed in the state shellfish records, and you see this very good straight line, extremely strong and statistically significant regression. So we now have the opportunity to use these instruments for a number of things, one of which is to actually try to give estimates of shellfish toxicity without the state having to do all of those kind of tests. So one vision of the future for us in monitoring these problems is to put an array of ESPs along the coast in key locations. So they can provide this data, the cell count data that can be assimilated into the models, like I showed you, to make our forcecast models better, but they can also give us estimates of shellfish toxicity at these nearshore monitoring stations to complement what the states are doing. So that's a big plus and a big advance. So moving quickly to the second instrument, it's called the IFCB, or the Imaging Flow Cytobot. And this is basically a submersible microscope. It goes underwater and it takes pictures of cells, very high resolution pictures. So here's another movie. This one I have to narrate. So we're going to open up the cover of the IFCB so you can see it. It's got this laser over here, you'll see in a second. We draw in a sample from the outside, and then we push it down through this flow cell. And as it goes through the flow cell, it gets hit, these particles and cells are in single file. They get hit with a laser. If it's a piece of dirt, it just gives off something called side scatter. If it's an algal cell, it gives off chlorophyll fluorescence, which is a red color. And that triggers a camera, which takes a picture of that particle. And that sounds complicated, but this instrument can do 10 images a second. So 1,000. I just took 10 images of cells like this one. And what you'll see in a second is that humans are not fast enough to identify them, so we have to teach the computer to it. And so computer machine learning then is what's needed. Here's just a few statistics over here. 8 to 10 images per second, 600,000 images in a day. Basically, humans can't deal with that. We usually joke at a university that's typically what we have graduate students do, but even graduate students can't do this. This is asking too much. So this is why we have to use automated species classification, just like on TV or in the airport where they're doing that facial recognition. It's the same thing being applied to cells here. It's not rocket science. So this is what we call a dashboard of output from that IFCB. It shows all the big cell types here and smaller ones. This is actually an Alexandrium bloom, all in this. And as you'll see, these are big Alexandrium cells, smaller ones that we know are gametes. These are tintinids and other organisms that are large. These are diatoms. There's many, many different kinds of organisms here. And one of the ways these IFCBs are being used, this one's in the Gulf Coast near Texas, but it could easily be used in Florida as well, is for early warning. They have put 3 ESBs out along the coast on piers that draw water up from below, and you can see they've had early warning of 7 HAB events. So the instrument sits there and is just taking samples, 3 samples an hour, 24/7, and then you can see that it picks up this organism, which causes this syndrome called DSP, and at this point, they alerted the managers, went out and tested, and they found things were toxic, and they've done that multiple times. This is Karenia, the species you have a lot of problems with, again, being picked up here. The instrument basically has thresholds set in it that if you get to a certain point, send out an email and text message to managers, to the scientists, saying you've got a bloom happening in this location. So it's being used for early warning very, very effectively. But I know some people here might be interested also in climate change and changes in pollution. Can you clean up the Indian River? So I stuck in a few slides here about that what we might call trend analysis. Let's look over long periods of time. What can this instrument give us? So here's an example of, think of this as cell number. It says biovolume, but it's the same thing. Over 6 months of time off of where Woods Hole is. And this is a measure of the total number of microplankton in the water, but the blue are this one class of algae we call diatoms. And you can see these 6 species are the ones that are largely responsible for those patterns. And that looks interesting, like, okay, we have something going on. But if we actually take it apart, we see that this organism blooms in January. This one blooms between February and March. This one between March and April. You can see how different all of these are. And that pattern that I showed you in the beginning is really the result of all of these different species that are called. And this instrument sitting out there unattended for 6 months, sending all this information back to someone's computer at the lab, everything's being done. It's an incredible way to monitor. And this is what I've been telling people here, that I think that you should have some of these in the Indian River. It's a great way, and this is where I'm looking that maybe we can help make that happen. So back to long-term monitoring, this shows a number of years, and this is one type of organism, a little blue-green alga called Synechococcus. These are diatoms again, but this just goes through 2009. But imagine we have this data through right now, more than 10 years, 12 years worth of data, and this shows you through these months of the year, the interannual variability in just that one group. It's an incredible data set to look at climate change or changes in pollution. You don't have some technician who's the one who's been counting your cells leave and go take a new job, and you have someone else come and try to learn how to count so that the data are questionable from year to year. This is being done the same software year after year after year. So back to our use of the IFCB. This is where we've been using it thus far in these small little salt ponds that I mentioned. And surprisingly, this is Alexandrium catenella. I didn't think this was really going to work, but it has. The reason I didn't think it was going to work is that these are all Alexandrium cells here. And we would somehow have to teach that software how to identify and distinguish all of these stages from the many other co-occurring things, that noise that I showed you in that other picture. So we have 5 different types of what we call singlet cells by themselves. These are doublets, cells that could be dividing, cells that could be fusing. We have cells that are infected with a parasite. You can see here. You can have quadruplets. You have these large planozygotes that we have here that are the result, right, the precursors to our cysts. And we train the software to do that and are getting incredible insights. And this is maybe a little too deep for some, but I hope you can follow the excitement of a scientist who for years has been studying an organism that has this complex life history. And now all of a sudden it's like we've got a window, we're looking into the bedroom. You know, we're in there saying we're peeping Toms. We are seeing what's going on in a way we never could before. So what do I mean? This is just an example of the bloom in one particular year as tabulated by the IFCB. Where, again, it's being done in volume. We've turned cell counts into volume. It's the same thing. So among the things we can observe here, this is now the volume of the singlets, those single cells I showed you. It can be multiple types, but any single cell of Alexandrium, what's its volume? And you can see that it goes up and down and up and down and up and down. And you see this is nighttime and daytime, nighttime, daytime, nighttime, daytime. And the first point to be made is that through this early stage of this bloom, the volume doesn't really change very much. But just when this bloom is going to peak, cells start getting smaller and smaller and smaller. And they get really small down here. And then what happens? They start getting larger. You don't see this up and down pattern anymore. It's just they're getting larger. So what's happened is this is where we are having basically mass gametogenesis. We're producing huge numbers of gametes during this end of the bloom. This is why the bloom ends. The cells have gone through that sexual transition where they make gametes that be all the small cells here that fuse together to make those large zygotes, which don't divide anymore, which then fall out as cysts. And interestingly, we get more than 90% of the population making these gametes in the field. When we do this in the lab, if we get 15 or 20% in our cultures, we're happy. So the sexual induction of this organism, by the way, you'll see a few mistakes here. We've had to rename it. It's now Catanella, but I'm not changing species on you. The sexual induction is far more robust in situ than in cultures. So we're seeing really what's happening. Now, let's take a look at that same pattern. And what else can we do? Because these cells are getting bigger and then smaller and bigger and smaller, what's happening is at nighttime, they grow and they get big and then they divide. And one cell makes two and therefore they're smaller. And they get too large and then they divide, large and divide. When that happens in this kind of phase division, you can actually calculate what we call an in situ growth rate. For the first time in a long time, it sounds like it's an easy thing. We can tell how fast these cells are growing in the field. Again, it sounds like we should have been able to do that, but any phytoplankton ecologist will tell you how hard that is to make that measurement. But we can do that with this instrument, with that equation, and what it shows us is here. Here's a growth rate. Just think of that as how fast do they grow, how fast do they divide. Here's temperature, 5, 10, 15, 20. These are some laboratory data where we took and brought cells from that area, grew them in the lab, and at 10 degrees, these are the various growth rates we got. At 15, we get this. These are the growth rates we're calculating in the field at those same temperatures from that IFCB data. And you can see that they're much, much higher. And I could give you other reasons to tell you, to convince you that that's not an artifact, but I don't have time, but believe me, we've looked into it, and we do believe those are true in situ growth rates. So what does it mean? That these cells divide 2 to 4 times faster in situ than in culture. So not only are they having a lot more gametes, a lot more sex, they are also growing faster than we thought. Let's look at how fast they're swimming. Another thing we can do is follow their swimming up and down. You can see this is the surface of the water here, and they migrate each day. They swim up to the surface, and then they go down, and they swim up, but they don't quite get to the surface. And we can actually measure the rate of that swimming, and basically we're getting rates of 15 to 20 meters per day, which, as you see up top here, in the lab and in tanks and so forth, we were thinking they could swim 5 to 10 meters per day. So what we've also seen in this is interesting here. Remember this pattern where they're sort of going up and down, and then this is the migration of those cells through time blown up. And what we see is that these cells are avoiding the surface here, which is one of the reasons they stay within this pond. But after the point where they have become these zygotes, they are swimming up higher and higher in the water column, which means that they're up near the surface here and can get washed out of the pond. Well, is that true? Well, this is where we took the IFCB, put it inside a jet yak, which is a kayak with a jet ski engine to it, and it's remote controlled. We can let it go out here into this shallow marsh system. So here's the pond. We did all sorts of surveys here with it, brought it out there, did all these surveys looking for what was out there, and what did we get? And now you've seen this type of dashboard before. But what you see here now is the only Alexandrium cells we're seeing are these big, big, big dark zygotes. These are all a different organism. This is something called heterocapsa, and this is yet another organism there. So the only cells leaving the pond are that late history stage. So what we're saying then is that two things. These cells swim and migrate faster in situ than predicted, and migration behavior varies among these life history stages. We never knew that before. So this has very significant implications in terms of the dispersal of the species. And the final little set of these slides, you're now familiar. This is the bloom. We actually had sort of 2 blooms this year. I won't go through all of this, but look down here. Here is where we actually deployed an ESP and an IFCB together. We used the IFCB to watch the cells, tell us what's going on, and then when we thought things were interesting and there were cells there, we would trigger the ESB and have it measure toxin. And this line down here shows you the toxicity. That's what this axis is. The toxin per cell of the cultures established from this very salt pond. And you can see it's in this fairly narrow range. This red line shows you the toxicity that we measured in the field. And it basically was much more toxic than what we thought it was. And I won't go into it, but we also have in situ growth rates that are going in here that, again, are faster. So what did we learn from that experiment was, first of all, that the toxin content of these cells varied more than 4-fold during the bloom. So the cells aren't equally toxic all the time. Sometimes they're really toxic, sometimes they're less toxic, but it was higher for the vast majority of the time than what we thought that those cells could produce. So once again, these in situ instruments are showing us things that are very different from what we thought was the case using our laboratory studies we've been doing and everybody's been doing for years. We also saw that this in situ growth rate varied as much as 4-fold over just a few days. Temperature would get cold, growth rate, they'd stop growing, they'd grow slowly. Warms up, they would grow faster. So we saw a lot of dynamics in that as well. Okay, the final little point here is that you've already seen the jet yak. We now are using a number of different mobile platforms for the IFCB and the ESP. Here you see the jet yak. I know that Harbor Branch here has a wave rider which uses wave energy to move autonomously through the ocean. The IFCB has been put on this and can be again robotically just move all over the place taking samples. We've put it on board vessels that can move rapidly and give us maps of organisms. I show you this because the ESP is now in its 3rd generation out at Mbari. This device here, looks here, this is now the ESP. So now it's gone from the size of a trash can to the size of a soccer ball. And it fits inside this vehicle, which is kind of cute here. Here it is, this wave's diving. Here's this little vehicle here, which can dive, come up to the surface, send its information, go back down. You can tell it where to go, and it's got an ESP inside it. So this dream that I showed you of what we think a future monitoring program should look like can change. Before I showed you either ESPs or IFCBs moored along a coast or situated along a coast to give you early warning, to give you data for bloom model assimilation and estimates of toxicity, but we also now can go mobile. That's sort of the next phase of all this. So we can have an array of moored and autonomous ESPs and IFCBs and other instruments providing real-time data on the distribution, abundance, toxicity, and even the physiological condition of these cells. So scientists are supposed to dream. This is mine, and I know the pieces are there. There's a lot of expenses, there's a lot of trial to go, but this is not too far-fetched, believe me. So my final thoughts for you. I hope you believe that the HAB problem, the red tide problem, is serious. It's a global threat to health and to coastal fisheries, aquaculture, tourism, water security, lots of things. And it's not going away. It's not going to change. It's been around for many, many years. It's worse now. I haven't gone into that at all. Worse for lots of reasons in most parts of the world, but it's not going to go away. So we have to manage it. Numerical models of HAB dynamics are advancing rapidly. You have some in this region, and they are providing forecasts at both weekly and annual timescales. But in all cases, the accuracy can be greatly improved through the use of this in situ biosensors that I've showed you. And 2 instruments, at least in the northeastern U.S. right now, show great promise. That's the IFCB and the ESP. But in addition to their use in providing data for the modeling and forecasting, they are providing insights not possible with traditional sampling and modeling approaches. That's what I meant about we're looking into the life history of this organism. We're looking into their blooms. We've got cameras right there watching it happen. It's an amazing new, it's a transformational technology. You hear about Amazon and others being transformational. I believe that about this technology. And in particular, what it's shown us is that the cells swim faster, divide faster, form more gametes, and are more toxic than we thought. And the bottom line is that if we're going to understand and forecast these HABs, here, Florida, New England, wherever, characterization of critical rates and behavior in these natural populations, basically, that's what we need. And those need to be generated whenever possible through in situ observations. Concurrently, the biosensors that we're talking about also can provide early warning information as well as cell abundance for the forecast, like we said. Many of the technologies and approaches described here can be used on Florida HABs, and that's my sort of what I'm saying to all of you and to my colleagues here at Harbor Branch. This is a wonderful opportunity for collaboration. I think there are things that can be done here to greatly expand your knowledge of what's going on, especially in the Indian River Lagoon, a very, very important site for HABs. And I hope that something may come of it and we could work together, and maybe I can come back and talk to you again. But thank you for your attention.